physiological or an ecological adaptation, which allows those individuals to move into that new habitat and essentially then diverge into two different species. However, the way in which species respond to environmental change is not necessarily always the same. And that's because a species' response to environmental change can be driven by many different factors. And those factors can include differences in species phenotypes, their dispersal capabilities, their ecological preferences, physiological limitations, or their life histories. And understanding how this variation in species responses to environmental change occur is critically important in terms of understanding and reconstructing the biogeographic history of particular communities, in addition to understanding how different phenotypic and ecological innovations have evolved in newly derived habitats. And these 2 points relate to the 2 areas of research that I'm going to talk to you about today. The first is looking at the biogeographic responses of organisms and how that relates to their environment. And the second looks more at the interactions between ecology, phenotype, and evolutionary history. And while these 2 areas of research are not necessarily mutually exclusive, my research so far detailing this first area is concentrated on understanding how the variation amongst species within a particular system in a comparative context. My research in this second area primarily has been dealing with the interactions between ecology, phenotype, and evolutionary history in driving adaptive diversification across closely related organisms. In a systems sense, I'm interested in understanding how these processes play out in desert and semi-arid habitats. Desert habitats are particularly interesting to me because they represent some of the most recently derived and extreme habitats that we see on Earth. And they're also expanding at a rapid rate with climatic change. In another sense, semi-arid habitats pose another interesting question to me in that these spatially and temporally, these habitats represent some of the most dynamic habitats that we've seen. And occurring on the edge of desert systems, these habitats have expanded in and out with glacial cycling, and consequently the spatial distribution of these habitats has shifted dramatically, and they've experienced a great flux in environmental conditions within specific areas. And how evolution plays out in these environments is particularly interesting to me also. And so this brings me to the first area of my research that I'm going to talk to you about today, and that's looking at the biogeographic responses of organisms and how that relates to their environment. My research in this area draws heavily from the methodological and analytical tools associated with the discipline of phylogeography. In a general sense, phylogeography seeks to take genetic samples from across the distribution of a species in order to understand the demographic and divergence responses amongst populations within a species. Namely, we seek to use genetic data to link the timing of particular events, let's say a demographic event like a population expansion or contraction, that may be associated with a particular environmental change event. We're also interested in understanding how different environmental events can be linked to the timing of population divergence amongst different populations within a species. In order to understand how these processes are playing out amongst members of a community within a particular region, we need to take a more comparative approach. And this discipline is detailed in comparative phylogeography, and one of the central tenets of phylogeography has so far been understanding the common evolutionary responses of organisms within a particular community to specific environmental events. And the example I have up here are the comparative phylogeographic population divergence patterns observed amongst species in the North American deserts. What we have here are the dotted lines that demarcate areas of divergence that have been seen in many different species that occupy this environment. However, simply focusing on the commonality of species responses, I think ignores the potential variability that can be seen within a system. I think what my work and the work of others has been showing is that the causes of population divergence can be rarely singular, in that many different species may have different responses in different areas. Namely that you may see spatial differences in the geographic position of breaks that are seen amongst different species. You may also see temporal differences in that a particular environmental event that's caused a divergence amongst populations of one species may not necessarily be the same event that's driven population divergence in the same area for another species. Another type of difference that we see is that you can have very different demographic responses to particular environmental events. One environmental event may cause one member of the community to expand while another member of the community may contract. And my research has looked at these kinds of questions in many different amphibian and reptile taxa. However, for the purposes of today's talk, what I'm going to focus on is some more recent work I've been doing in the coastal reptile community of southwestern Australia. This community occurs in a linearly distributed habitat from the south coast of Western Australia through to the mid-western coast. It's restricted to this grey area here, which are a series of coastal sand plains and sand dune areas. Now, this area is also one of the most diverse and considered a biodiversity hotspot for two particular groups. Plants, in particular, if you go to this particular region here in Kalbarri, you'll find at least 2,000 flowering plant species. One of the other groups that is particularly diverse for this area are the reptiles also. And in order to look at what might be the factors driving population divergence in this system and thereby leading to this incredible diversity that we see in the system, I wanted to look at a series of sympatric species that occurred across the distribution of this habitat. And more importantly, what I wanted to do was include a series of species that had different life histories and ecologies. So what I did was I selected a small fossorial skink, a semi-arboreal gecko, a large fossorial skink, a terrestrial skink, and a terrestrial gecko. And there are a number of hypotheses that have been discussed in terms of explaining diversity in this system. Many of them relate to historical processes. The first is vicariance. Vicariance is often considered the process by which an environmental event here, an example is a mountain chain rising up, which then splits this species into two disjunct populations. Habitat stability is another hypothesis that's been used to explain the diversity in this system. With sea level changes associated with glacial cycling, this system has seen an enormous spatial shift in the distribution of coastal habitats. In that when sea levels have dropped, the coastal sandplain habitat extends out almost up to 160 miles and then contracts back. And so one hypothesis is that you might have areas that have remained suitable across time housing genetic diversity of populations and thereby having driven divergence amongst those different populations. And one of the great examples linking habitat stability to the driving divergence amongst the community comes from Arnacarnarvon Craig Reeds. Here they looked at this spatial distribution of Atlantic forest habitats that was predicted at last glacial maximum, last glacial minimum, and present day. And the areas in black demarcate those areas that are predicted as suitable for Atlantic forest at each of those time periods. And what they found was that genetic diversity and divergence amongst populations was directly linked to these areas of constant habitat stability. The third hypothesis that I wanted to consider is not often considered in the discipline of phylogeography and relates more to the distribution of habitats, and that's related to habitat connectivity. And if you imagine here we have patches of habitat that are suitable for our particular purple lizard species here. These three patches of habitat up the top are sufficiently close enough to one another such that they allow gene flow between them. Whereas this population down here is separated from this block up here by a sufficiently large area of unsuitable habitat such that there's no opportunity for gene flow amongst these areas. And so I wanted to test if this hypothesis could explain the variance that we see in population divergence in our reptile species. So what I did was I took some genetic samples from across the distribution of each of my species. I collected some sequence data from mitochondrial and nuclear sequence markers. I then used the spatial points here to generate some species distribution models from some environmental data that I collected from standard GIS sources. I then had my 3 different hypotheses that I need to test. So I set up a series of grouping variables from both the literature and from these data here, and I'll explain how I did this. The first is our hypothesis of vicariance. And what I wanted to do is group populations either side of known vicariant barriers in both singular barriers and combinations of the 3 different barriers that we know to exist in this habitat, and see how that grouping variable could explain variation in genetic divergence. The second grouping variable that I set up was habitat stability. And what I did was I projected species current distributions onto last glacial maximum climates and looked at those areas of habitat that were predicted as being suitable in both time periods. And those areas here are depicted in this particular species as these black areas, whereas the grey areas are predicted as being suitable currently, but were not predicted as being suitable during the last glacial maximum. And therefore I grouped populations according to their stable versus unstable status. The third hypothesis I want to test is looking at habitat connectivity. And what I did was I used a current species distribution model to look at the sufficiently contiguous areas of suitable habitat that were separated from one another by large areas of unsuitable habitat. And this unsuitable habitat areas or the breaks in suitable habitat are depicted here by the dotted lines. And so I use these 3 different grouping variables to test how much of the genetic variance, the variance in genetic divergence could be explained by each of these variables in an analysis of molecular variance. And if you're familiar with an analysis of variance, in this particular case what I'm doing is I'm using a dependent variable, which is my genetic divergence, and I'm using the grouping variables to see what proportion of the variance each of those can explain for each of my species. And so what I have here are my 3 different hypotheses and the proportion of variance explained by each of those grouping variables across each of the species. I have a range of different values here for vicariance because if you remember, I had both singular barriers and combinations of the different barriers that exist within the species distribution. And what I want you to note from this is that despite some high proportions of variance explained by vicariance in some of these species, habitat connectivity explains the highest proportion of genetic variance in every single one of these species. And what I think is this commonality of a response actually belies the incredible variation that we see amongst our different species. And what I have here are the species distribution models and areas of unsuitable habitat demarcated here for each of my 5 species. And there is incredible variation in both the number of potential areas of unsuitable habitat within each species, in addition to the spatial position. So if we focus on this one here, this species doesn't have it, it's shifted further south, it's further north here, and it's somewhere in the middle here. So there's variation in the spatial positioning of these areas of unsuitable habitat. And so, given this result, what I wanted to do was think about what might be the abiotic factors that are underlying this variation in habitat suitability, potentially explaining some of the variation we see in genetic divergence in this system. And so what I did was I took my genetic data again, I created a genetic distance matrix from the multiple loci I collected. I then used the spatial distribution points that I have to extract environmental data for each of these individuals. I created a series of abiotic variables from each of these. The first is climate scores that I created by reducing the climate data down to the first two principal components. Another variable describes soil types across the distribution of the system. I then wanted to take my dependent variable, genetic distance, and see what proportion of the genetic variance could be explained by my abiotic factors across species. And so what I found was that a significant amount of variation could be explained in the genetic divergence in every single one of the species by both climate and soil. And in fact, soil predominantly was a factor that in some cases was able to explain 40% of the variance, and 47% of variance in genetic divergence within some of the species. And so, what does this all mean for understanding the causes of variance in species responses to environmental change? Well, I think the fact that you have things like soil and climate that are able to predict patterns of genetic variance and variance in genetic divergence have strong implications for suggesting that species' physiological limits and their ecological preferences may be playing a role in driving divergence amongst populations. Soil and climate are critically important for determining thermoregulation in reptiles, in addition to climate being important in habitat distributions and things like that. And so, in a broader context, I think that this might be suggesting a role for local adaptation as a driver of population divergence. And I think this is an incredibly exciting avenue for future research. I'm now going to move on to my second area of research I'm going to talk to you about today, the interactions between ecology, phenotype, and evolutionary history. And for this part of the talk, I'm going to focus on some research I'm currently undertaking on this group of sand dragons from Australia. In addition, I'm going to talk to you about some of the projects that I'm currently pushing underway in my lab currently on Galapagos tortoises. So far I've been talking to you about this interaction between ecology and evolutionary history. But there are also critically important interactions between species ecology, evolutionary history, and their phenotype. And some great examples of this are where you get an environmental change that drives a new ecological opportunity, driving the diversification of species. And we see many examples of this in nature. Some of the great examples we have are the Darwin's finches, where species have been able to modify their beak shapes to take advantage of abundant food resources across different islands in the Galapagos archipelago. Another example of this are the enormous lizards, where the radiation across the Caribbean has repeatedly evolved different phenotypes to take advantage of the different structural habitats that are available to them within those islands. And while these are classic high biomass systems, one of the other big systems that's been inferred to have evolved a lot of adapted radiation are the Australian deserts, and close to my heart, being Australian. Australia is one of the most diverse reptile systems in the world. We have an incredible number of reptiles. We are also 70% arid. And many of the hypotheses surrounding the derivation of this diversity involve some form of adaptive evolution. And while all of these hypotheses are out there, very little evidence quantitatively looks at exactly when and how these species may have been able to first get into the arid zone and then diversify across the arid zone. And so to look at this a little further, I took a suite of 11 species that cover the distribution of mostly the southern semi-arid and arid habitats. These species are largely allopatric and cover the distribution, this distribution here. They also occupy an incredibly diverse array of habitats, from sand plains and sand dunes that occur both inland and in coastal environments. And as we can see, There's also a big array of different colours of sand and soil types.